the difference between when it comes to AAV mediated gene therapy and then ex vivo lentiviral gene therapy. What would you say are the diseases that are a better fit for each modality and why?
Yeah, sure. Just to quickly explain the difference. So with AAV gene therapy, your drug product is a virus that contains a therapeutic genome. So you manufacture that virus, put it into a vial, and then the treatment modality is that you infuse that virus into a patient, and the viruses, latch onto cells and deliver their genome into the cell. That genome makes its way to the nucleus and then expresses an RNA that gets translated into a protein. So in that case, your drug product is the virus itself. For ex vivo lentiviral gene therapy, the drug product is actually the patient's own CD34 positive hematopoietic stem cells that have been altered through the use of a virus outside the patient's body that are then delivered back into the patient. At AVROBIO, our methodology is that we mobilize those CD34 positive cells, extract them from the patient's blood, send them off to be manufactured at a facility using lentivirus to deliver the genome into the cell. When those cells are transduced, they go through the final QC release process as the final drug product, and then they go back to the clinical site, and those cells get infused into the patient, where they take up residence in the bone marrow, give rise to progeny, and then those genetically corrected progeny go all over the body. So the question of which modality is better is pretty controversial. There are advantages and disadvantages to both. I would say that there are probably some diseases that simply can't be treated by ex vivo lentiviral gene therapy, and there are some diseases that can't or shouldn't be treated by AAV gene therapy. A good example, I think, is a disease that I worked on when I was at Sarepta Therapeutics, Duchenne muscular dystrophy. So the Duchenne muscular dystrophy is caused by a defect in the dystrophin protein, which is the largest single gene in the human genome. It's huge. It takes over 16 hours for the RNA to be transcribed from the gene, and the final mRNA molecule has 70 exons in it, and the protein itself is, a huge protein. And this protein is part of a complex that connects the cytoskeleton of muscle cells to the extracellular matrix, and it makes connections with a ton of other proteins within that complex. So one of the mechanisms of action for ex vivo lentiviral gene therapy is that the corrected cells in the patient's body will transfer, in many cases, RNA and protein from themselves to neighboring cells. So it relies primarily on cross-correction, unless the disease happens to occur in one of the progeny of hematopoietic stem cells, in which case you're directly correcting the disease. But if you're relying on that sort of transfer between neighboring cells, that works very well for a lot of lysosomal storage diseases and other diseases that involve soluble proteins that can transfer easily between cells, or even membrane-bound proteins that can transfer between neighboring cells. I think it's very unlikely that you could get that to work very efficiently for Duchenne muscular dystrophy, because the protein is so large. It has to be very carefully chaperoned as it's being made into position within that structure, where it makes connections with other proteins. If you just make that protein without any sort of appropriate chaperones, it's going to fold in weird ways, and it's never going to be functional. So in that particular case, it's very important to actually make the protein in the muscle cells in order for the expressed transgene protein to be functional. But there are other cases where AAV is probably not the best modality, and ex vivo lentiviral gene therapy is really the way to go. Gaucher disease, which is a disease that AVROBIO is working on, is a good example. In Gaucher disease, macrophages are one of the primary cells that are affected by the deficiency in the GCase enzyme, and macrophages descend from CD34 positive hematopoietic stem cells. So by doing the ex vivo lentiviral gene therapy, you're essentially correcting one of the cells that is most affected by the disease.